Hello all and welcome to the 30th session of the Global Ocean Oxygen Network webinar series. I'm happy to be with you here today. My name is Jeremy Sterling from the Ocean Science section of IOC-UNESCO, and I will be moderating the webinar today. On behalf of the co-chairs of the Global Ocean Oxygen Network, the GO2NE, I'm delighted to welcome you to this session. We are joined today by Simone Moretti of the Max Planck Institute for Chemistry in Germany. And I'm sorry to report that our second speaker, Hussein Nabhani, has just yesterday let me know of an urgent matter that has prevented him from attending today. So instead of our usual hour, we will have a slightly shorter session. We will have about 20 minutes of presentation, followed by a 10-minute Q&A session. And if you would like to ask questions at any time during the webinar, you can use the Q&A button on your Zoom bar. If you have it set up the same way I do, it's a little button at about in the middle of your screen at the bottom that says Q&A. If you have any additional questions or you would like to chat or have any logistical issues, please do use the chat function. I see already some people in there. Welcome. And I will shortly hand over to Simone, but first, a very brief introduction. So, Simone has studied geology at the University of Bologna for his bachelor's degree in Italy and earth science at ETH Zurich in Switzerland for his master's. In 2022, he obtained his PhD from ETH Zurich and the Max Planck Institute for Chemistry in Mons in Germany. Now he is continuing as a postdoctoral researcher at the Max Planck Institute for Chemistry, where he is funded by a DFG grant, the German Research Foundation, I believe. Simone will tell me if that's correct.
Sure.
Indeed, thank you.
That's correct.
To reconstruct anthropogenic impacts on nutrient cycling and ecosystems in the Southern Ocean. So he will present today on some recent work, the presentation titled The Future in the Past, Studying Past Ocean Oxygen Using Nitrogen Isotopes and Body Size of Fossil Foraminifera. Simone, welcome. Thank you very much for joining us today. The floor is yours.
Thank you very much. Okay. Okay. Can you see my screen? Hope.
I can see it well.
Okay. Let me just move the one. Okay, perfect. Well, so thank you all for being here, and thanks a lot, Jeremy, for this introduction. So today I will be talking about how we can use geochemistry, and in particular nitrogen isotope, as well as metabolic modeling of body size of microfossils to reconstruct past states of ocean oxygen. And in particular, past state of ocean oxygen in warming events in the past, in the geological past, and how we can perhaps learn something from these past warming events to better understand the future of ocean oxygen as the climate continues to warm. So it's well known from a wide body of literature that the detrimental effects of global warming on ocean oxygen, as well as marine ecosystem, as well as human activities that depend on them. However, when we compare those studies that are well known to the studies that deal with the past of ocean oxygenation, we see something that may perhaps raise some eyebrows, or in any case, invoke some curiosity, because the warmest period of the Cenozoic appear to have been associated with more oxygen, at least in some part of the ocean. And as I will make a sort of a quest for me in this talk to convince you that these titles are not necessarily contrasting, I will be mostly talking about our most recent work from the Paleocene, Eocene thermal maximum. So models tend to agree that as the climate continues to warm, the vast majority of the ocean volume will progressively have less oxygen by the end of the century that it has now. However, some simulation experiments that have attempted to run future states of ocean oxygen for several centuries and thousands of years notice that this deoxygenation trend may eventually come to a halt or even reverse to higher value in one case for sustained emissions. However, there are very few of these modeling exercises, and the uncertainties are large because these simulations rely on our capacity to correctly quantify marine biology and the degree of respiration in the ocean interior, as well as the degree of ocean ventilation from high latitude regions. And while the models are getting better every day, the uncertainty still remains. So an alternative, and I would say complementary approach to this, is to look at the past, and in particular at past warming events in which the effects of global warming on ocean oxygen already unfolded. Therefore, we can learn get some insights on what the future of ocean oxygenation may be on the long term. So in this figure, you see here the evolution of sea surface temperature from paleoceanographic reconstruction over the Cenozoic. That's what geologists call the last 65 million years. And you also see this in the same scale with the projection for the end of the century under two different scenarios. So you can clearly see that the Earth has been going through several climatic shifts, but one that particularly strikes the eye is, in this compilation, at least, is the Paleocene thermal maximum, or PTM. This event occurred around 55 million years ago, is considered by many to be the fastest pre-human warming event of the Cenozoic, and as such has received a lot of attention from geoscientists as an interesting study case for understanding the response of the ocean and the Earth's climate to rapid warming. So is the PTM really like a perfect study case? Unfortunately not. So at most, the PTM had about a fraction, let's say a quarter of a degree Celsius per century rate of warming, which is at least an order of magnitude slower than the current rate of warming that we're facing. Nonetheless, there are some similarities that we can look at. For example, the magnitude of the warming during the PTM is similar to what we may face if we fail to cut our carbon emissions and continue to emit CO2 at current rates. So we know without any doubts that the causes of ongoing global warming is linked to fossil fuel and our addiction to fossil fuel, whereas in the PTM it was caused by natural release of greenhouse gases in the ocean atmosphere system, such as volcanic activity and methane released from the seafloor. The impact on life that are projected to occur in the future includes a loss of habitability and a polar shift of many species, and perhaps even a mass extinction. Now, this finds some similarity also in what happened in the PTM. There is evidence for tropical extirpation of species that move toward the poles, and it also has the PTM had the largest extinction of deep ocean organism of the Cenozoic. And for deep ocean organism, it was even larger than the extinction that caused the demise of the dinosaurs 10 million years before this event. So is the PTM a perfect study case? Unfortunately, it is not, but it does share some similarities that may help us to understand the future of ocean oxygen on the long term under worst case emission scenarios. So what we will discuss here is how did the ocean oxygen change during this PTM event? And also we will try to address to which extent these findings could be relevant for understanding the future. So to address this question, we will make use of foraminifera nitrogen isotopes, and I will explain later on what we mean with that, and also metabolic modeling of foraminifera size, as well as comparing our result to Earth system simulation with high emission pathways. So nitrogen isotope can be used as a proxy for ocean oxygen. In brief, nitrogen has two major isotopes in nature. One is nitrogen 15 and one nitrogen 14, the latter being the most abundant. And the different abundance of these two isotopes in any natural sample can be expressed in delta notation. And as you see here, to keep it simple, like larger value of delta 15N reflect a larger proportion of 15N in any sample. So most biologically mediated reactions discriminate between isotopes, a process known as isotopic fractionation. That is because usually the lighter isotopes react slightly faster than the heavier isotopes. And the rate or the ratio of the different rates of reaction between the two isotopes is usually described by a fractionation factor, epsilon, And the larger the epsilon, the larger the so-called isotopic fingerprint of that process. So the larger difference of rates of reaction. So one of these biological reaction is denitrification. And this is where oxygen comes into the discussion, because denitrification is the microbial reduction of nitrate to N2 and can only occur in waters where oxygen concentration are below 5 micromolar. In the water column, this condition are only met in the East Equatorial Pacific and the Arabian Sea. Now, water column denitrification has a strong isotopic fingerprint, therefore a large epsilon, which leaves nitrate enriched residual nitrate from denitrification enriched in nitrogen-15. And you can see in the map on the right that the Pacific is isotopically very heavy because of this. But you can also see that while denitrification only occurs in the easternmost region of the Pacific, the isotopic signal of this nitrogen-15 enrichment is seen throughout the tropical zone. And that's because the assimilation of nitrate and the remineralization of the organic matter that is produced from it spreads this isotopic signal well beyond the spatial extent of the oxygen deficient zones. So the take-home message from this slide is that we can use nitrogen isotope to track denitrification, which in turn is strongly linked to ocean oxygen in the East Equatorial Pacific. Now, how can we go back in time? So foraminifera look like this, are unicellular organisms that produce a calcium carbonate shell. And at the end of their life cycle, they sink at the bottom of the ocean. And this is how they look in their fossil states. And they get preserved in sediments. Fortunately, we have ways through ocean drilling to access this sediment and retrieve the sedimentary cores and extract the foraminifera that are preserved in this sediment for millions of years. But you may ask, okay, why foraminifera? And foraminifera have organic linings in their carbonate that are mostly proteinaceous material. And it has been shown, for example, by Abby Wren in 2012 in Danny Sigman lab that the isotopic composition of these organic linings correlates very well in modern foraminifera to the isotopic composition of the nitrate in the pycnocline where these foraminifera live. And in this way, they offer us a sort of a time machine to reconstruct the isotopic composition of the nitrate before the beginning of instrumental observations. Of course, this relation works only in oligotrophic regions, so regions where nitrate is fully consumed, so that we don't have any other nitrogen isotope fractionating processes. In case, for example, of non-oligotrophic regions, the enhancement of nutrient consumption would also act to elevate the delta 15N in ways that have nothing to do with denitrification. And that's why we need to choose sites that are in oligotrophic regions. However, I'm showing you these slides because this concept will come in handy in a couple of slides, as you will see. So nitrogen isotope can also help us understanding the degree of nutrient consumption in non-oligotrophic regions. So how do we measure this isotopic composition in forams? And by the way, it's not only a foram thing, we can also measure it in corals and other biominerals such as diatoms. Well, we need to first dissolve the biomineral matrices and release this proteinaceous material in solution. We can then oxidize this proteinaceous mixture to nitrate, and finally feed this nitrate to a culture of denitrifying bacteria that we culture ourselves for converting this nitrate to N2O gas. Finally, this N2O gas can be measured in a mass spectrometer coupled with a custom-made gas bench, and we can get, therefore, our isotopic composition. So this is a very laborious method. It's called the oxidation denitrifier method, but it allows us to analyze very small amount of nitrogen down to 2 nanomoles with a very high precision of 0.2 per mil. And I'm reporting here some reference in case you're interested in learning more about this methodology by looking at the recording of this presentation later on. So the study sites that we will discuss in this presentation that we explored are the one that you see here. So the circle are the one where I will discuss about the nitrogen isotope evidence, and the square is also where we will have foraminifera metabolic modeling that I will introduce later in the slide. So to jump to our results, we see in this figure on the x-axis a timeline of a relative age of onset of the PTM. So the 0 is the onset of the event, the negative values are kiloyears before the onset and positive value are after the onset. So in the lower panel, you see the well-known rise of sea surface temperature from a global compilation of previous published data. And in the upper panel, you see our forum-bound result. So what you can see from our oligotrophic site is that there is a sharp drop of delta 15N that occurs in both the Atlantic and the Pacific, but the largest change by far is in the Pacific basin. Now, based on what we introduced before, a decline of delta 15N implies a decline also in water column denitrification in the Pacific. And that means in our interpretation, that the best interpretation that we can give to this signal is that reduced rate of denitrification imply a more, let's say, an increase of oxygen in the water surrounding the ODZs during the PTM warming. Now, however, there is one site in this compilation that we studied that does the exact opposite, and that is site 865. Now, this site was located in the located in the equatorial Pacific. And you can see that the rise in delta 15N cannot be explained by denitrification alone. And we tend to think that this site records neutron replete condition. This would explain the lower delta 15N values before the onset of the PTM, and the rise in delta 15 will then reflect a more complete nutrient consumption. Now, this is important for us because this enhanced degree of nutrient consumption, it's something that we can link to water column stratification in a warmer climate. And in fact, this is not in disagreement with what CMIP6 simulation predict under SSP5 8.5 scenario that you can see here on the right from previously published result. In this case, I'm citing Kwiatkowski et al. 2020. And you see that this stratification in this model has the implication that also reduce the supply of nutrients from below to the surface. And reduced supply of nutrients implies reduction of productivity. So this consumption of nitrate in the PTM may also provide us with a mechanism to explain why oxygen may have increased in the water surrounding the disease in the first place. Because reduced nutrient supply, reduced productivity would also reduce the oxygen demand in the ocean interiors. And this seems to be occurring to some extent also in the simulations that I'm citing, where the East tropical Pacific somehow seems to be refractory to the deoxygenation that is seen in most of the subsurface. And in this case, I'm plotting the depth of 100 to 600 meters. Of course, I'd love to hear more of the thoughts of modelers, because here I'm citing modelers, but I'm not a modeler myself. So I would love to have a confrontation between this data and the simulation. But a small recap of this first part is that we see a reduction of denitrification rate during the warming in the PTM. And in parallel, we see this evidence of nitrate depletion in the tropical Pacific. And we interpret that this decline of denitrification is linked to a reduced oxygen demand in the ocean interior that is linked to reduced productivity because of stratification in the tropical Pacific. Now, I find this very exciting because it goes in the same direction of the prediction for the end of the century under worst case scenarios. But the question that now we can ask ourselves, are this finding only relevant for the ODZ regions, which is only a small portion of the ocean volume, or do they speak for a wider part of the tropical ocean? And this is because the nitrogen isotope only track denitrification, as we've seen, which is a very specific process that happens only in a small part of the ocean. And it's for this reason that we turn our attention to a different approach, an independent approach, that is metabolic modeling of the size of foraminifera. So foraminifera are ectotherms, so that is organisms that are not capable of regulating their own body temperature. The metabolic ratio is a parameter that describes the capacity of an environment to meet the metabolic needs of an organism. And it's quite intuitive that this metabolic ratio, phi, which is described as the ratio of supply and demand, needs to exceed a certain critical value that needs that is necessary in order for the needs of the organism to be met, and therefore they can fulfill their metabolic needs to ensure feeding and reproduction. And this metabolic ratio has been described by Curtis Deutsch in 2015 in a science paper. So that's quite a reference to look for you if you're interested in deepening your knowledge on this topic. What's interesting is that this metabolic ratio links body size of organism B and environmental temperature to environmental PO2. Now, there can be two sensitivities that can be extracted from this equation, from this metabolic ratio equation. That is the so-called temperature size effect. And for the vast majority of marine ectotherms, warmer ocean implies smaller foraminifera, and therefore a dwarfing reaction to reduce the metabolic pressure by a warming effect. But there is also the so-called oxygen size effect. And also lower amount of partial pressure of oxygen will induce a dwarfing of foraminifera for the same reasons. So how can we parameterize for foraminifera that lived million years ago? So what we did is compiling respirometry experiment based on modern foraminifera, and then compare this outcome with the theoretical prediction. So where do we expect the parameters that we extracted from these current respirometry experiments on living species based on a wide range of metazones? And we see that, for example, the parameters that we can extract fall exactly where we would expect them to be on theoretical grounds, which give us some confidence that we can use modern respirometry experiment to apply this in the past to species that unfortunately don't exist anymore. So we have this equation that links body size and temperature of foraminifera, but you may ask, how can we also get this information? Fortunately for us, this information exists already because Oceanographers have been compiling size of fossil foraminifera during the PTM, as well as paleo temperature, using a different set of approach that, for the sake of time, we cannot explain here, but the reference, you will find them in here. So what's important is that you see that the foraminifera that lives in the deeper layer of the ocean, so those are the benthic foraminifera, show dwarfing. So they get smaller during the PTM warming, but planktonic foraminifera that live in the uppermost layer of the ocean shows a rise in size. And this is quite unintuitive because, as we've seen in the previous slide, the warming of the PTM would tend to induce for most aquatic ectotherm a dwarfing, and instead we see an increase in their size. Now, if we apply our metabolic model, we see an interesting pattern arising, because we see that the increase of size of planktonic foraminifera implies an increase of partial pressure of oxygen in the water surrounding the habitat of this foraminifera, for this planthic foraminifera. Whereas for the foraminifera living at the bottom of the ocean in the same size, we see a reduction of partial pressure of oxygen. And so we have the coexistence of a signal of tropical enhancement of partial pressure of oxygen during warming, but also a deoxygenation of the deep ocean. So a small recap of what we've done here so far is to use metabolic model of foraminifera size to infer past changes in ocean PO2. And the parameterization is based on modern respirometry experiments and input variables from previous published foraminifera size and temperature. We see a signal of oxygenation that is located at a site that is distant from the oxygen deficient zone. And so this is quite important because it means that the oxygenation that we saw with nitrogen isotope is not something that is only linked to the eastmost region of the Pacific, but can also be seen toward the more central part of the equatorial Pacific. And interestingly, we see a complete decoupling between the tropical upper ocean and the deep ocean, which is by far the largest volume, that instead shows a deoxygenation with warming. Now, finally, I just want to discuss a little bit the modeling. So what we try to do, now we're looking at the same data, but with a different perspective. So now we're plotting depth here, so the habitat of this forum, or the paleo habitat of this forum, and the delta PO2 in terms of relative change of partial pressure per degree Celsius. And as we discussed, we see an increase of PO2 in the upper layer and a decline of PO2 in the deeper ocean. Then we use a general circulation model forced with PTM temperature field, and we try to model what is the response of PO2, and we see a remarkable similarities of the simulation and the actual observation, paleo observation. Interesting, we see that when we use the same model, but now forced with SSP5 8.5 scenario, we see a similar pattern arising with this tropical oxygenation occurring, but also deoxygenation of the largest volume of the ocean in the deep. So this observation may help explaining some biotic pattern that were long known in the PTM, and that is, as I introduced in the beginning, there was a largest extinction of deep organism during the PTM, but this extinction of deep organisms somehow was not coupled with an extinction of organism living in the upper layer. And this has remained puzzling for a long time. So this remains speculative at this stage, but perhaps the deoxygenation of the deep ocean may have been responsible for this huge extinction event, but perhaps the tropical oxygenation in latitudes that contains the highest amount of biodiversity may have at least provide some buffer that may have helped avoid a mass extinction, at least during the PTM. And another key take-home message, I think that there is a strong consistency between simulation and this for the future and the paleo data. And again, I would like to show these slides from another, from always the same CMIP6 paper I cited before, where the tropical Pacific seems to be refractory to deoxygenation during warming events for the end of the century. And in the simulation here, these patterns occur. So again, I would love to talk more about this with modelers and hear their thoughts about this and how we can maybe work together to bridge more the gap between paleo data and modeling. But the summary that I want to give you is that two independent proxy for the PTM argue for more oxygen in the tropical subsurface during this warming event, and that this rise of PO2 interested a larger fraction of the tropics, not just the spatial extent of the disease, and that, quite excitingly for me at least, this pattern of pressure of oxygen change across the PTM are consistent with the projection for the SSP5 8.5 scenario and CMIP6 simulation. And finally, and this remains, of course, speculative, the observed divergence of oxygen trend in the PTM may explain the minor Planktic extinction in comparison to the largest benthic extinction of the Cenozoic. So I hope I'm not over time. I'd like to just to advertise, if you're interested in learning more about past state of ocean oxygenation, there is a working group called PO2, and that's part of the PAGES Past Global Changes consortium, and there will be a workshop in November in Bristol where we will discuss these topics and many more. So you can scan this QR code if you're interested, and it will be available in the recording as well. And if you're more interested in this work, you can find this here. So thank you very much for your attention, and I'm happy to take your question.
Thank you so much, Simone, for an excellent presentation.
Thank you.
I'll start with the Q&A. Thank you all attendees for listening. Remember, you can use the Q&A box down at the bottom of your Zoom window, near the middle, round to the left, for any questions you have of the speaker. But maybe to kick things off, I actually had a quick question that came to me. Maybe it's not very difficult, but are there any analogs for when you were talking about the decoupling of the deep ocean and the shallow waters in terms of ocean oxygen and ocean oxygen during the PETM? Do we see that at other times or even in other models?
We do. So interesting. We don't even have to go back in time that much. So there is some, I'm thinking about this paper from Sam Jakar where he compiled different proxy for ocean oxygen during the last glacial maximum. So we're only talking about 20,000 years ago. When humans were around, and there is some divergent trends also in surface and deep ocean oxygenation. So we see the same we see this decoupling sort of what happened in the upper ocean and what happens in the deep ocean.
Do you think it may be of a similar reason?
Well, so I think it may be difficult to just make a direct parallelism because of different boundary conditions and different processes, but I think definitely biology, so the respiration of organic matter in the tropical surface can explain the same observation of divergence between the upper ocean and the deep ocean, which on this timescale is still dominated by changes in ocean ventilation. So another thing that is quite difficult to predict on the long term with models is how the ventilation from high latitude region would respond to global warming. So whereas I'd love to hear the opinions of modelers on this, it seems like we are doing maybe a better job in understanding how the respiration in the upper layers of the ocean affects oxygen more than how the change in ventilation on the long term will affect deep ocean oxygen.
That maybe leads into another question then on collaborations between paleoceanographers and the modeling community today. I wonder, from your experience and your work on this, what thoughts do you have on advancing the use of paleoceanography, maybe in conversation with modelers or with contemporary oceanographers or even with decision makers today?
Yeah, so that's an excellent question, and I'd love to see more like modeling effort in like trying to reproduce past observation. Like, can we use a model to actually generate the same trends that we see for the past? Like, can we validate our current models using past data? That would be a great way to both like validate the paleo work and also validate the models, and perhaps also making paleo work more visible to a larger community. I think that's something I would definitely love to explore more in the future, to work more with modelers in this direction. And paleo scientists, like any scientist, should do a better job, I think, in communicating science more effectively and in simple terms, and for sure trying to make it in a way that is more easy to digest for policymakers.
Indeed.
Yeah.
All right, so we have a question from the chat. Yeah, I think it's a good one, because of course it's very regionally and even locally specific sometimes, deoxygenation. But so from your talk, we talked about deoxygenation in different strata of the ocean, but also in different regions of the ocean. Does this mean that in some parts of the ocean, oxygen concentrations are rising and some parts oxygen concentrations are decreasing?
Well, so this is problematic because there is some observation that, in fact, oxygen deficient zone may be expanding, so that even in the tropics, there is deoxygenation. So to my knowledge, there's not that strong of an observational evidence that today we see an increase of oxygen yet. So what we need to be careful on is when we are looking at the problem of timescales. So I think at the moment, we are seeing the fastest effect, that is the effect of solubility. So as the ocean keeps warming, there will be less oxygen in the water. And so I think we are definitely seeing everywhere this effect. The effect of global warming on biology and ocean ventilation is something that will take decades and centuries to kick in. It doesn't make it less important. It just means that we're not able to see that effect so clearly yet with observation. And that's why we need to rely on models, and we need to rely on observation from the past. So to my knowledge, we don't see a clear oxygenation signal happening right now, but based on our findings and what simulations say, it may be that we're going to see it in the next decades to centuries.
Thanks so much. Just a quick comment. I see someone, Urias Taylor, yes, indeed, the recording will be available later on YouTube. But we have some more questions here. Do you think that CO2, the size approach that you mentioned, so the metabolic ratio, can be used to reconstruct the expansion of tropical OMC during the Quaternary? We just touched on this a little bit, I think.
Yeah, so in fact, we are going to try to do that with Curtis. We want to team up and have a project just on the last glacial cycle. We think this approach can be applied pretty much on timescale that involve thousands to tens of thousands of years. I have some concern for applying it on timescale of millions of years, because then maybe evolution may make things more complicated, because when we are seeing a long drift in size of foraminifera, for example, we just don't know if it's maybe really an environmental thing or an evolutionary thing. But on short time scale, where let's say evolution can be ruled out, for example, the PTM, if you believe the shortest estimate of onset was only like 2,000 years, and I say only 2,000 years from a geologist's perspective, but if we look at the deglaciation, we're talking about 10,000 years or less. So on this time scale, I definitely think we can use the metabolic modeling to reconstruct past changes in partial pressure of oxygen for both the oxygen deficient zone, but also the deep ocean, using benthic foraminifera. Okay, and other ectotherms. So not only foram, could be ostracods, or could be other ectotherms.
Okay. Oh, also yes, I'll pass this on. Thank you very much. I see in the Q&A an offer of collaboration. Okay. All right, then maybe the last thing we do before we sign off, you mentioned two methods to use forams as a proxy for ocean oxygen conditions. How did you decide on these two methods specifically? Well,
so the nitrogen isotope for us was easy, because that's what we do pretty much in the Max Planck Institute as our main analysis. And this proxy is quite unique because it really specifically tracks denitrification. So a problem we have in paleoceanography is that there are a lot of variables that may affect one proxy in a way that sometimes it's not always easy to really pinpoint, to reconstruct that variables that we really want to reconstruct. And nitrogen isotopes are quite strong in that, because in oligotrophic regions, they really track the global budgetary change between inputs and outputs in the nitrogen cycle. And the major output in the nitrogen cycle is denitrification. And denitrification happens only in low oxygen conditions. So it's a qualitative approach, because it doesn't give you a number. It doesn't tell you this was how much oxygen you had at any given time. It just tells you that less denitrification was by necessity linked to more oxygen. But then the reason why we chose to go with the other extra approach is because the other approach can offer you a quantitative tool, because if you can have a metabolic model of foraminifera size, you can come up with estimations of what actually was the partial pressure of oxygen. Like, unfortunately, the weakness of using a metabolic model is that you need to rely on correct parametrization. And I think this is something that we need to do more work on in the future. We even need to do more experiments with trying to do foraminifera culturing and really see if we can build a larger data set that we can have better and better parametrization, but also going out at sea, collecting forams, measuring environmental temperature and PO2 and foraminifera size in situ, right where they live, not only from culturing, and calibrate a relation also with that. So these two approaches for us were different but very complementary. One is solid but qualitative, or the other one is a model but gives you numbers.
Okay, thank you. No, I think we may be discussing that approach again, possibly, hopefully soon on the webinar series. I know there is some very good ongoing debate around the use of the metabolic ratio, the metabolic index. So hopefully we will have some good discussion on this in the future. But for now, I think it is almost time to sign off. I will share this last slide with everybody that I hope you can all see. And I will thank you so much again, Simone, for your presentation, and for all of you today who have joined us for this 30th session of the Global Ocean Oxygen Network's webinar series. Before we go, I'd like to highlight some of the ways that you can learn a bit more and stay in touch. the oceanoxygen.org website with the link on the left-hand side of your screen here. This site links to all past webinars and to the Global Ocean Oxygen Decade newsletter on ocean deoxygenation. The site's feed on the main page is also updated regularly with the latest scientific articles on ocean deoxygenation. You can also find out more about GO2NE, the Global Ocean Oxygen Network, and GO2, the Global Ocean Oxygen Decade, and all of the activities under their auspices via these links to the IOC-UNESCO web pages at the bottom left here. And so, switch windows. Thank you all again. Thank you for attending. Thank you, Simona. We hope to see you all again next time for our next webinar in August, and be sure to follow us if you would like via the social media links. You can use this QR code to sign up to the GO2NE mailing list, and you can use this email address to email us with any questions you might have. We will see you again in August. This webinar will be posted on YouTube in about a week. Have a pleasant day or night wherever you are connected. Thank you so much again. Goodbye.
Thank you. Bye-bye, everyone.